Good afternoon, everyone, and welcome to the fourth episode of Ewing's U. I am Piero Spada. I'm going to be your host today, and I'm one of the co-founders of Little Warrior Foundation and dad to 12-year-old little warrior Lucy. Today, we are thrilled to be hearing from Dr. Ramon Sun. A bit about him. Dr. Sun is a newly appointed endowed chair and associate professor of biochemistry and molecular biology, and also happens to be the director of Center of Spatial Biomolecule Research within the College of Medicine at the University of Florida. He recently made his transition from the University of Kentucky, and prior to that received his PhD in cancer biology at the Australian National University, followed by postdoctoral training at Stanford University. Lastly, Dr. Sun is also a member of Little Warrior Foundation's Medical Advisory Board. As we'll see here today, Dr. Sun specializes in using cutting-edge tools to quantify metabolites, which are signaling molecules that can sometimes be used as cellular energy sources. Specifically, his characterization of glycogen stores across many cancer types has led to a unique and potential dependency by Ewing sarcoma cells. More on that later. In his spare time, Dr. Sun is an avid dog lover who cares for 4 dogs ranging in size from a small Pomeranian all the way up to a Doberman Pinscher and Greyhound, and is also father to an active 3.5-year-old son. But before we kick things off with Dr. Sun, I just want to let everybody know and remind you that Ewing's U is a webinar and podcast production of the Little Warrior Foundation. We are a national childhood cancer foundation with a laser focus on finding a complete and permanent cure for Ewing sarcoma. And we were founded in April of 2020 and have granted out $1 million in high potential therapies for increasing survivorship and decreasing toxicity. We very much so rely on the support of our communities of the big and little warriors who are joining us on this call and across the country. We really appreciate all of our community support to continue our mission so that we can bulldoze our way to a solution and continue this vital work. And with that, before I hand it off to Dr. Sun, I'm going to pass it off to Emily to introduce herself and a couple of housekeeping items.
Thank you, Piero. I'm Emily McFadden, one of the other co-founders of the Little Warrior Foundation. And real quickly, just wanted to take you through some housekeeping items before we begin. Dr. Sun is going to talk for about a half hour, and then we'll leave time for questions at the end. Please use the Q&A function at the bottom of your Zoom chat to submit questions. And then Piero and I at the end will ask those questions of Dr. Sun. As always, we can't discuss specific medical cases or provide medical advices in the context of this webinar. And then, I think we're going to cover some pretty dense material today, and I want you to know that this is going to be recorded, and we will have the transcript and the recording available for you to re-listen to if you miss anything. So with that, I will kick it off to Dr. Sun.
Well, thank you guys so much for the invite, and thanks Piero for that awesome introduction. I just wanted to say that I stumbled upon Ewing sarcoma research, and it is really like meeting people like you and the rest of the Little Warrior families that keeps the fire going for me to just continue to work on Ewing sarcoma and continue pushing along the line of research to try to eventually bring something from the bench to the bedside, hopefully in the near future. Having said that, my talk is going to be a little science heavy. I apologize up front if it was not directed to the right audience. Blame Piero. No, I'm just joking. But feel free to ask any kind of questions. If anything in the science is not fully understood, just let me know, and or email me later. Happy to chat with anyone online or offline. So I'm just gonna get started. Like Piero said, I just moved over from Kentucky to Florida. As you can see, I'm still at my own house. My lab is not set up yet, and half of my people are still in Kentucky doing work. So hopefully the transition will finish in about a month or so. But in the meantime, just a lot of writing and a lot of housekeeping stuff. So my lab, as a research group, really is interested in how do we connect genotype to phenotype. And what I meant by that is when you look at the genetic makeup of all of us, the DNA. So the genetic makeup, humans have 20 to 25,000 genes, but when you look at other organisms, dogs have 19,000 genes. A mouse who's, who I frequently do experiments on, they also have 25,000 genes. And a plant that's, on the back, in your backyard or in your front lawn, they have greater than 40,000 genes. So it's hard to tease apart organismal complexity just by looking at genetic material alone. Human and dogs and mice have over 90% shared genetic sequences. It's really hard to basically just tell what sets us apart from dogs or mice just based on looking at the genetic material alone. So when you expand from that, DNA gets translated, gets transcribed, into RNA, and RNA gets translated into protein. And and every single protein has a metabolic function. So these proteins either enzymes that continue to do metabolism in our body, or they become structurals where they form long chains of structural components that lines the outside of your cells, your bone, or any kind of your body's component. And it is this protein to metabolite interaction or metabolism that's normally what give us what we call like organismal complexity of human physiology. And it is really the control of how protein interacts with metabolite that really gives us that interactome network. So for example, a single protein can have multiple different ways of getting regulated inside of a cell. So you have phosphorylation, acetylation, glycosylation, myrcylation, alkylation. There's endless possibilities of how we can regulate a single protein to do its function. But the other interesting thing is all of our proteins are ubiquitinated, which means it's getting destroyed and resynthesized within our cell. And 80% of all of our protein in our body, human body, does a complete turnover in about every 72 hours or 3 days. So, if you really think about it, we're really essentially a brand new human being every 3 days, which is kind of cool when you think about it that way. So it is this complex interactome network that's beginning to break. And when this, any protein that breaks, it perturbs this metabolic network. And then that's what happens when you lead to metabolic diseases. So when I was a graduate student and most of my postdoc, we're really cool that we're living in an age where, rapid technology innovations are being applied to do academic research and having these, amazing data being translated into the clinics. So when I was just a trainee, we used to study transcriptome, proteome, and metabolome in a variety of different ways. But one thing's certain is we generally mix up our organs. So I've actually used a physical blender to blend a cow liver before because it's so massive. So you mix up everything that you want to study inside of this organ, and then you kind of study the metabolic profiles and phenotypes and genotypes. So one of the major problems of doing this approach is the lack of account for cellular or heterogeneous metabolism or responses. For example, if you're looking at a tumor, you have the cancer cells, you have the stroma, and you have the necrotic regions, right? So if you pull all of these regions together, for example, if something is decreased in the tumor, but increased in stromal and necrotic regions, which doesn't mean it's not important. Not everything happens inside of a tumor. It's pro-tumorigenic, has to happen inside of a tumor. It can happen in the surrounding microenvironment. So if you pull everything together, this is actually a piece of Ewing sarcoma, a human tumor actually, so it's kind of cool. So when you pull all of these regions together, a lot of these exciting phenotypes that we should be going after or we should be investigating suddenly come out with a net zero. And when you see that net zero as scientists, you generally just let it go and you don't go down that route anymore. So it's kind of cool that we should be able to tease apart all these cellular responses and rather than mixing them all together. And this piece of tumor is no bigger than an M&M. So just what I mean, like, you know, it's a very small cancer, you know, not Ewing in general, but, you know, if you even from such a small piece of a part of a tumor, it's already have this many regions. How are we really teasing apart what is really tumorgenic or tumor regressive? So in the last decade, I think for those of you who's really following academic research and science, there's a lot of advances in terms of how we study a lot of these. So a lot of these things, you know, single cell RNA-seq, and I believe single cell RNA-seq has been applied to Ewing sarcoma as well. What you see here is all of these different unique sub-cell types getting clustered by single cell RNA-seq. But what they don't have is a spatial of where in this solid tumor are these cell types present. Then you come into the spatial proteomics analysis, where you have all of, you can do single cell proteomics, and now you can look at cell types and how they are, you know, represented next to each other. And then you can do spatial metabolomics, which is what I'm really interested in and specialize in, where you can see where all the different cellular layers and where all the metabolism occurs in all the different cell types. And in the future, my lab is really focusing on, really focusing on integrating a lot of these different next generation techniques, moving them together and really using them to tease apart specific cellular and regional metabolism with unique diseases. And if you're like me, you think all of these technologies cool, you know, we're not the only people who are doing it. You know, Nature and a lot of different articles are beginning to highlighting the innovative and the importance of doing single cell metabolomics analysis by multiple different organizations. Just give me 1 second. Sorry about that.
Dr. Sun?
Yeah.
This is Emily. I think this audience would benefit from kind of just zooming out one level on what spatial metabolic mix is, kind of at a really kind of a high level, and how it's different from other types of research.
Yeah, so that's spatial metabolism. It's really, so if you, as I progress, it's really the spatial metabolism is teasing out these differences versus looking at this part of the interrogation. So majority of the current work in the lab currently, you're looking at this bar graph over here where there's no change because it's from a mix of material. Where spatial metabolism is different is where you can look at each individual regions uniquely separate from all the different other regions, where there's no response of mixing or any other kind of responses. I think it will become more clear as I start doing there's a lot more actual examples of this.
Perfect. Great, thank you.
Forward, in the later on part of the slide. I just, you know, wanted to so like the way we do this is really just cut a piece of tissue, coat it with some ionizable material, and then put it in this laser ionized this really cool machine. So you kind of coat your tissue with this and like special chemical material that helps the ions ionize. And this is where the laser ionization happens. So you put this in the machine, laser will hit the slide, and you have the empty slide, and you have the brain regions. And you can see as the laser tracks through the different regions, you have, and then it's starting to collect a lot of metabolic information as it hits the actual piece of tissue. And the end results is what I'm like mentioned earlier. It's all these different metabolites that's uniquely localized in all the different regions, which all of these information that that would be lost when you do a pooled analysis, when you put everything together. And you can, you know, represent it in like a spatial setting where you overlay all the different metabolites that's happening. So what I mean by this is like you really need to tease apart what happens in each regions inside of a tumor before you can really come up with usable or effective therapeutics. If you're just looking at it a bigger picture kind of a way, we're losing a lot of scientific information that just rushing in to do therapeutic development. A lot of the times, a lot of drug fails because we're missing a lot of the spatial information. So we can either do these by looking at single metabolites overlay. Again, these are all human tumors, right? So that's where the cancer is. You can see like a lot of the metabolism actually does not happen inside of a tumor. So this is where a cancer is. And these are all stromal regions. And you can see this is a colon cancer. You have those little hairs coming out of it that's attached to your colon. You can see a lot of metabolism doesn't actually happen in the cancer. A lot of these surrounding cells, they act as supporting structures that support cancer metabolism and facilitate cancer growth. So not everything that's pro-tumorigenic or pro-cancers are directly happening inside of a cancer. And you need to identify all of these combinationary kind of a point together before you really can design a very effective therapeutics in our mind. Because if you want to target as red, for example, that's very high in this special form of lipids, it's not happening in the tumor, right? So if you just hitting a lot of these cells around the tumor, you're really not impacting how the tumor is growing. It may be helping the tumor, but you're not really directly hitting the tumor, which is what anti-cancer therapeutics aim at doing. So based on this, we can profile a lot of different metabolites within a cell. That goes from glycogen, oxphos, which is like ATP bioenergetics, lipids, as well as like structural carbohydrates. So that's the outside in the extracellular matrix components that's really make up the different parts of a cancer cell. And today we're gonna go a little bit more in depth into like our role in glycogen metabolism and what have we been doing with that. So just even my worst part of my undergrad experience is study biochemistry and glycogen metabolism, even as an undergrad. So hopefully this does not put all of you to sleep, which is really boring biochemistry. So glycogen is how our body stores glucose, which is mostly the stuff that we eat. So when you eat a big bowl of pasta, for example, during lunch, maybe a lot of you just had lunch, so a lot of glycogen synthesis happening right now in your liver. So our body uses the liver to store all of our circulating glucose and suck it up into the liver as stored as glycogen when you overeat. But during a period of starvation, for example, if you miss the meal, that's when glycogen breakdown occurs, and it's really just aimed at leveling your circulating glucose. And inside of a cell, you can find glycogen kind of in multiple different cellular locations, in the nucleus, in the mitochondria, in the cytoplasm, and ER. And glycogen is degraded through multiple different mechanisms inside of our cell, right? So we have lysosome as well as glycogen phosphorylase degradation. So different route leads to different roles for glycogen. And glycogen is also heavily phosphorylated, again, decides which route a cell takes. This is important to know. Again, scientific background is important because it will come in handy later when we talk about Ewing sarcomas, because we have to know how glycogen is utilized and different route and how it's controlled and been utilized before we can actually design a therapeutic strategy, because we can't just hit one or the other one and ignore the other, because one of the things doing research, we all come to the realization that a cell is very, what they call metabolic fluid. It's almost like a leaky pipe with multiple different points, and you block one, it just comes out the other end, and you block the other end, it just comes out another one. So the metabolism of a cell is very metabolically fluid. So you have to find out the exact mechanism how things is utilized before you actually hit it, because otherwise all you're doing is redirecting the nutrient pathway, and it really doesn't do anything to what happens inside of the cell. So to do this, we designed a brand new technology, something that hasn't changed in the last 80 years, really, in the lab, how we measure glycogen. And that's part of the reason why we're missing a lot of this, Ewing sarcoma. It's because the technology hasn't changed in 80 years. Very much similar to the treatment Ewing sarcoma patients. It hasn't really changed. The technology hasn't changed. Therapeutic treatment hasn't changed. So because of that reason, we developed this new multi-imaging technique that we can actually starting to visualize where glycogens are localized in a boosted sensitivity that is like we're guessing 10,000-fold more sensitive than what the previous technique is, which is the PAS staining. So many of you might have heard the PAS staining before, because that is one of the clinical diagnostic features of Ewing sarcoma. So when you resect the tumor, you put it under a microscope, and if you do a PAS staining, and if it's red, it's being shown that given the diagnosis of Ewing sarcoma. So with this, we can suddenly starting to do a lot of different cool analysis in different tumors and all the different tissues. And we can starting to look at how big the glycogens are, how much phosphorylated they will be, and really starting to understand what is underlining the accumulation or the lack of glycogen when you look at a tissue-specific setting. With this, we can suddenly starting to see glycogen in many unexpected tissues. So you have glycogen in the lung, glycogen in the brain. You can beginning to see that all of the different structures are very different among different human organs. So our body, not just liver, stores glycogen. And many other tissues like your brain, the lung, kidney, they all kind of store glycogen, and they all use them for different purposes. And to this end, we don't really know what each organ's metabolic demand is for glycogen. That's some of the things that we're trying to actively work on and discover in the future. But one of the cool things about having a new technology is that we can profile a lot of different tumors, different subcell, different tumor types. And you can see here prostate, 2 different lung cancers, and then Ewing sarcoma on the far right. And looking at their glycogen accumulation, and glycogen and Ewing sarcoma is included is because we're interested in this PAS positive phenotype in Ewing sarcoma. And you can kind of see that Ewing sarcoma accumulate glycogen by far the most amounts compared to all the other kind of human tumors. So you can see that it's the Ewing sarcoma is probably 10 to 50-fold higher than prostate and this lung squamous cell carcinoma, and it's about 1.5-fold higher than the lung adenosine carcinoma. Glycogen is actually important in this tumor as well, but I'm not going to go into it. But really, we're just going to try to figure out, is this a viable therapeutic target inside of Ewing sarcoma? And again, you can kind of see, because of spatial technology, you can see that this is where the tumor is, and this is the stromal region, the fat region. There's a fat pad here that's attached to this human tumor, and these are all muscle fibers. And you can see that glycogen is very uniquely localized inside this tumor, and it's not there at the other regions, which suggests that it's a very specific Ewing sarcoma tumor feature. So I think usually I do a little introduction on Ewing, but I really, for you guys, you need no introduction. But we all know how devastating this disease is. This is, it's extremely heartbreaking to see our kids have to go through surgery and chemotherapy, and it's even harder as a parent. I can't even imagine how parents would deal from losing a child from Ewing sarcoma. So that's part of the reason that we decided to work on it. And 2 really cool clinical phenotypes that you often see in at least children's Ewing sarcoma cases is one that everyone kind of know, that is the EWFLE1 fusion oncogene, and the 2 is the PAS positive histology, which looks like that. So this is what clinicians, pathologists use to diagnose with the help to diagnose Ewing sarcoma. We know that they form these big giant red lumps inside the tumor. But no one, we know that clinicians has been knowing about this big red patches that you can see over here inside of Ewing sarcoma for decades. I mean, since it was discovered, we knew that these big red patches are forming inside the tumor, which is why I was super surprised about 3 years ago when I started working on Ewing sarcoma. And then I asked around, literally no one knew, one, what those red patches are. They kind of guess it's glycogen, but no one really know. And 2, no one's really looked into it, because this is such a striking phenotype inside of a Ewing's tumor. No other tumor, very few other tumor have this phenotype. So you're looking at hundreds and hundreds of different tumor types. You can count the number of tumors with these specific red patches on the number of your hand. So there's very few tumors that have this phenotype, but it just really surprises me that no one's really going after this phenotype and figure out why it's accumulating and is it a therapeutic intervention or target. So like I said, majority of the ES tumors has been focusing on EW1, and I think me talking to a few people alone, I know that there's a number of drugs that's trying to hit this we call it oncofusion genes, and I think there's a lot of good preclinical efficacy that's been showing that it's very effective at targeting, showing a lot of tumor regression. So hopefully we're going to see a lot of advances in this in the next couple of years and help a lot of children. And ideally, if this drug is working, I would be gladly to be put out of a job for looking into glycogen, if this drug works. But before this is proven to be a cure, there's no point of stopping Ewing sarcoma research, and it's better to have multiple shots on goal, what we call multiple shots on goal, so that just in case one fails, you have multiple different backup options to be able to follow up if your primary option's failing. I'm not saying EW1 is failing. I'm just saying like we should have backup options and not put our eggs into the single proto-oncogene egg basket. I wish with all my heart that this works and everyone else can be cured of Ewing sarcoma. This is honestly best case scenario. So let's move on to the PET staining. So this is what when your children have Ewing sarcoma and their cancer gets cut out, they get sent out for analysis. This is what the pathologists see. So the doctor look at the slide and you see all these little circular pink granules that's pretty much everywhere inside of a Ewing sarcoma. These arrows are pointing this, and this is what I mean by PET positive staining. This is being used to diagnose for the diagnoses of Ewing sarcoma in the last couple of decades, and to the fact that the doctors nor researchers know what they are or what do we deal with it, right? Is it a therapeutic target? So PES, just the background, it sees all forms of carbohydrates. And if you don't know what carbohydrates are, honey, maple syrup, they're all just kind of carbohydrates, long chains of sugars joined together. And carbohydrates by nature is sticky. So if you don't know what carbohydrates do inside of a cell, just imagine put a drop of honey between your finger and try to stretch it. That is exactly what carbohydrates are doing inside of a cell, just how it's doing. It's very sticky, it's gluing cells together, it helps support it in place. So when we first worked on this project, we're really curious about just what exactly is PET staining. It could be any of these carbohydrate-related molecules. We guess it's glycogen just because it's shape, but there's really no way to know until recently that we got our technology in place. So here we got a number of patient Ewing sarcomas from shoulder, chest wall, rib, abdominal, and bladder. And you can kind of see that glycogen is heavily accumulated strictly inside the Ewing tumors. And what's more interesting is that when you look at how glycogen is accumulated, so that the structure of glycogen, the tumor actually looks very much like the muscle that is surrounding it. So now that you can kind of beginning to tease this apart, and you can see this is one of the shoulder Ewing sarcoma patients. Here is the tumor region. Here is the muscle that's got resected at the same time when you take the tumor out. And then you kind of know that, hang on a second, and then when you compare it to tumor versus necrotic regions, you see how the tumor is exclusively in the tumor, but also the glycogen is exclusively in the tumor in this case, but the glycogen is also heavily accumulated in the muscle regions surrounding the tumor, which actually give rise to the interesting hypothesis. Like, look, Ewing is probably somewhat similar to the muscle cells surrounding it than other tumors of the similar subtype. They share very similar glycogen characteristics with the muscle cells surrounding the tumor. And then you can more or less confirm this, so you can get the mesenchymal stem cells that is give rise to all the connective tissue such as the muscle, the bone, and they actually contain a lot of lot of glycogen when you compare it to like a Ewing's tumor. So this actually gives us the hypothesis that, you know, tumor cells, Ewing tumor cells probably arise from these progenitor cells that give rise to all the bone and muscle that's surrounding a normal tissue. And therefore, you know, when as a scientist, you always, when you have two different phenotypes, EWFLE1 or glycogen, are they connected, right? So are they connected? So when we did that experiment, so we put EWFLE1 in these are kidney cells. So if for example, if this is the driver of glycogen accumulation, if this fusion oncogene drives glycogen accumulation, you should see that if I put this oncogene in a different subcell types, it would drive the glycogen accumulation up much higher. But what we don't see is that this oncogene is not a primary driver. Specifically over here, you see that the EWFLE1 is not a primary driver of glycogen, which tells you that they came from independent responses. So you probably have the high glycogen phenotype in the progenitor cells, such as mesenchymal stem cells, and then you have a secondary hit, which is your EWFLE1. So you have the combination of the two that give rise to Ewing sarcoma, rather than the oncogene shows up and then it drives glycogen and then it drives tumor growth. So they're kind of independent events, which means that you can kind of hit them as individual events in terms of therapeutic targeting. And like I said, glycogen is a fuel in your liver, right? So when you overeat, glycogen accumulates, and then when you during fasting, glycogen gets broken down. It's really just a metabolic reserve. So you can kind of see it's doing very similar things in terms of Ewing sarcoma. So if you look at the number of cells when you take away glucose, so we grow these tumor cells inside of a dish and we give them a media full of glucose. But then when you take them away, you can kind of see that the Ewing sarcoma is very resilient when during a nutrient deprivation. So after you take glucose out of this nutrient, Ewing sarcoma is very resilient for up to like 3-4 days without glucose because the glycogen is being utilized. But if you take a lung cancer, for example, that doesn't have a lot of glycogen, the moment you take glucose away from the cells, they all just kind of die. So you kind of know that when you look at lung cancers, glucose is absolutely important because if you take it out, they die, they don't survive. But when you look at Ewing sarcoma, you take away glucose from them, you take away their nutrient choice, they can still survive because they have large amounts of glycogen that's built into the store. And you can see that it's slowly releasing this glycogen. The glycogen store goes down slowly, but in other tumors, it completely disappears after 2 hours. So in a lot of the cases, we're current thinking is that high glycogen is really just like how liver is functioning. High glycogen in these Ewing sarcomas really acts as a metabolic fuel reserve to sustain tumor growth, and in future cases to sustain metastasis. So when a cell breaks down to go move to a different location, the glycogen help facilitate that whole process by providing a lot of fuel or nutrients for the cell themselves, and to continue to divide and metastasize. So with that in mind, we really wanted to know, is glycogen a therapeutic target, assuming it is a fuel depot for the cells? So we have two different modalities. One, this is a research tool, and then these are both research tools, right? So they're not therapeutic tools, but they give us a window into whether we can continue to develop therapeutic agents against glycogen. So one, you have this CRISPR knockout. So CRISPR is a way to knock out glycogen synthase. So glycogen synthase is the single only enzyme in a cell, including Ewing sarcoma cells, that makes glycogen. So you kind of either knock it out, or we have a small molecule inhibitor called Guaiaco. So this is actually a compound that's heavily concentrated in whiskey. Not advocating for whiskey consumption, but it's present in very low amount that gives whiskey the cool aromatic taste. But in much higher concentrations, it actually blocks glycogen synthase. So when you look at both of these approaches, So first of all, when you look at the CRISPR knockout, when you ablate a Ewing sarcoma to be able to make glycogen, and this is how we test the tumor growth. We put them in the mice on the flanks, and we can measure how fast the tumor grows inside of a mouse, and you can see that's a normal Ewing sarcoma that's growing, and it grows really fast even on the flank of a mouse. But when you knock out this glycogen synthase, which is responsible to make glycogen, the tumor essentially completely dead. It lost its ability to grow in a mice. And you can kind of see a very similar responses by us injecting this research grade compound that when you inject it into the mice, it has a very similar effect where it just kind of reduced tumor growth or tumor burden by these mice quite a bit. So this gives us a lot of hope, like, okay, glycogen, we know that the past positive stainings are glycogen, and we can pharmacologically manipulate glycogen synthesis to lower that amount of glycogen. And by doing so, we can significantly lower tumor growth in these nude mice. So this gives us a lot of hope to continue to working on this drug or this enzyme as an inhibitor to really, move this into the clinics in the near future, hopefully. Before I finish my talk, one of the things what I would like to highlight is this enzyme called AMPK, which is AMPK activated protein kinase. So AMPK is a master regulator that regulates a lot of these different cellular metabolisms. But what matters in the case of Ewing sarcoma is metformin. So we all know, and I don't know if your child was part of the metformin trial. That was a multi-site trial. So there was a metformin trial for a lot of different refractory solid tumors. And Ewing sarcoma is part of this. And one of our collaborators, Lars Wagner, is actually one of the co-lead up in Duke University. So there was a number of children that was part of this trial. And this is really want to highlight the importance of lab research is to really, we need to understand the molecular mechanisms or cellular mechanisms of why something works before we give them to our children, because if we don't, it might be potentially a waste of time that we can be trying other things. So one of the things that what happens AMPK is glycogen is a potent inhibitor of AMPK. And that's what your metformin is trying to activate. So when glycogen is present, it shuts down AMPK activity like this. So it shuts down all the AMPK activity, and it overrides any kind of activator activating products of metformin. So metformin may be useful in a lot of different tumors that doesn't have glycogen, but at least from our lab-based research, that based on our knowledge in how glycogen works inside of a cell, metformin wouldn't be a very useful therapeutic agent for kids that have Ewing sarcoma because of the heavy amounts of glycogen that's present inside of that tumor. So in a lot of cases, I think getting rid of glycogen in Ewing sarcoma cases would actually benefit the metformin efficacy. So, I don't know what I think the trial is ongoing, but it's coming to an end. And I think, at least early reports based on just direct communication with the clinicians, that metformin really didn't have a really, synergistic or additive effect to Ewing sarcoma patients, which, knowing what we know now could be a reason why glycogen could be part of the reason why our kids are not benefiting from metformin from a cancer regression perspective. With that, I think I like to stop my talk right here. I think it's about 35 minutes. I'm a little bit over, but I'll just skip the next slide, and would like to thank everyone in my lab. Lab is rapidly expanding. We have a lot of different funding agencies. I wanted to highlight St. Patrick's Foundation, V Foundation, and Raleigh Foundation, who support most of the Ewing sarcoma work. So they are very pediatric cancer-focused foundations that fund a lot of laboratory research, as well as junior faculty. And with that, happy to take any questions.
Thanks for that, Dr. Sun. That was fascinating on many levels. I'm going to give Emily some time to delve through the questions in the Q&A box. But I had a couple questions, and I was wondering if you could kind of allude, or let's focus on the translational aspect of these therapeutics. Can you talk about the other diseases like Pompe diseases and others that are heavily reliant on glycogen stores, and where those therapeutics are at in the clinical trials now, if you know whether you're inhibiting glycogen synthase or stopping its degradation? Can you talk a little bit about that?
Yeah, totally. So the most well-known disease that is getting treated with a FDA approved therapeutics is the Pompe disease. So they're formed glycogen aggregates. So in those diseases, the glycogen physically cannot be broken down. So there's no fuel perspective, right? So the glycogen is not being utilized. It just keeps getting bigger and bigger and bigger, and it's blocking a lot of cellular processes. So Pompe disease uses this, they call it acid alpha glucosidase. It comes in, it's an enzyme replacement therapy. So this enzyme comes in and clear the glycogen, and actually shown pretty good improvement. Like the patient is not cured, but they ended up living longer. Their physical motor neural responses are much better. They can walk longer. They have a lot more energy from a glycogen storage perspective. And then, among myself and colleagues, suddenly there's the debate of, is Ewing sarcoma glycogen storage disease? And if so, how are we going to treat it looking at a glycogen storage disease? And we do also work on, I don't know if so Maze Therapeutics actually works on a glycogen synthase inhibitor that actually is clinical grade. By clinical grade, I mean that is actually very potent, low toxicity, and they've tested in humans. And if you guys Google Maze Therapeutics, they just got approval for orphan disease status, which means the door can be open very soon for a lot of different diseases using that drug. And they're going through 100 volunteer phase 1 clinical trial to assess actual toxicity. So with that in mind, we think glycogen is a therapeutic target. How you're hitting it matters, because if you hit it with a enzyme therapeutics, right? What it does is it breaks apart these big glycogen to many, many smaller pieces. And in the case of glycogen storage, in the case of Pompe disease, that is good, because once you break it apart, it can be just digested. It's the big glycogen that is the disease driver. The cells are holding this big glycogen in, and because it's keep making bigger and bigger, it's not being utilized, the cell shuts down. But in the case of Ewing sarcoma, we've got to realize that you can think glycogen is a rechargeable battery. If you consider glycogen in Ewing sarcoma as a rechargeable battery, what happens when you break that resource down? You're just giving the cells more resources. It's a big ball of glucose sitting inside of a cell that's getting slowly broken down to support tumor growth. But if you come in too quickly to break this cancer down, to break this glycogen down, all it does is you providing more glucose to the cell to continue to divide. What we really need to do, this is again needs to be scientifically designed, is that we need to stop glycogen synthesis from the very beginning. And that is why we chose to hit glycogen synthase, because we have access to these enzyme therapeutics, and we never tested those in Ewing sarcoma cases, because we know that based on a battery model, if you actually hit it with the enzyme therapeutics, glycogen is going to get released. The cells are actually going to be happier. So believe it or not, I know that cancer grows, but Ewing sarcoma it's still a sarcoma. They're not in a very energy favorable environment. You could make them grow even faster by giving them more fuel, because there's no vasculature. They're surviving under odds because there's low oxygen, low nutrients. If you have something that have big glycogen and you're helping the cells to break down the glycogen, they're just gonna grow faster. So what we really need to do is just nib it at the source of glycogen synthesis, which is we really need to kill glycogen synthesis from the very beginning, and don't just see a phenotype and treat. We don't want to treat the symptom. We want to treat the disease. We don't want to treat the symptom of having large glycogen inside of a cell, because large glycogen have different disease drivers. In Pompe disease, it's because it's just getting too big, it's blocking everything. In Ewing sarcoma's case, it's because it's acting as a battery to store. So if you cleave this, it's going to get released and continue to add on a lot of different cells to give it more energy store. So I think the opportunity is there, but different glycogen storage diseases also have different target entry points and therapeutic entry points and windows. So we all kind of need to figure out what the actual disease driver is, and rather than just hitting that glycogen that's inside of our cells.
Oh, thank you for that.
We have a question here asking if this is a really popular topic in the Ewing space.
Yeah.
Would patients benefit? Does your research suggest that patients may benefit from extreme low carb diets, like a keto diet, to limit the amount of glycogen in the body?
Yeah, okay. So that's a really tough question, because I was literally just having another conversation with another colleague the other day. How do I put it? So if you look at in the case of athletes, who's actually doing ketogenic diet, ketogenic diet lowers liver glycogen, but it doesn't do much to muscle glycogen. Your muscles still have a lot of glycogen, and the reason is, for those of us who believes in evolution, we're evolved to run away from predators. Like a lion shows up around the corner, we need to be able to take off. And to be able to take off, you need energy. And what do you need? How do you store that energy for that sudden burst of activity? You store in your muscle. So to deplete muscle glycogen, you don't go ketogenic. Ketogenic depletes liver glycogen. It doesn't touch muscle glycogen. To really deplete muscle glycogen, and you see in my talk, we saw that Ewing sarcoma is very much similar to muscle glycogen. It doesn't look like liver glycogen, right? So, professional athletes who does a lot of, sprinting and running, they have lower muscle glycogen. That's one way of depleting it. But again, coming back to the ketogenic diet thing, there's a lot of benefit of eating a ketogenic diet just from a low carb diet for both adult and kids from a health perspective, like, because you're no longer eating garbage. You pick and choose fresh food and you cook it yourself. So those alone should have benefits for families, for kids to try. But I don't know if it would benefit Ewing sarcoma from that perspective, from a glycogen perspective. My philosophy for my own kid is like, what's the worst that could happen? I'm giving him a healthier diet, I guess, from that perspective. I think it's okay, but I can't tell you yes or no the ketogenic diet would in any mean affect Ewing sarcoma glycogen.
Great. So when a kid wakes up from surgery and wants a donut and nothing else, it's all right.
I think so. I wouldn't. That's only gonna hit your liver glycogen. I think we're looking at a much deeper problem here, just because it's accumulated in the muscle and it's a lot harder for your body to control from that perspective. Yeah, but from that perspective, yeah, like a donut, I don't think it's making the cancer grow faster. It's got other underlying mechanisms.
Thanks for that, Dr. Sun. That's a tough question for sure.
Yeah, it's a tough question. Yeah.
Yeah, I want to focus again more on the targeted therapy potential of this. It sounds like you're of the mindset that this is, if it's going to make it to the clinic, it's going to have to be in almost like a 1-2 combo with another targeted therapy, right? Because you kind of highlighted that Ewing sarcoma, we know, is driven by this EWS-FLI1 oncogene, which is independent of glycogen stores. And then you've also highlighted that the PAS positive hallmark of this cancer is highly glycogen dependent. So given that, is there a theory as to like when you would time that out? Like if you're, I guess to me, if you're blocking glycogen synthase in the mesenchymal cells, which are the precursors, does that happen? Is it almost like a maintenance therapy, or how do you see that moving forward?
Yeah, so a lot of factors goes into that. We don't really know how toxic inhibiting glycogen synthase is. The current data does suggest that it's the only bad side effect is you lose a little energy. It doesn't like have the same effects as chemotherapies, right? So if that's the case, is a big if, and we're hopeful if that's the case, it is possible as a maintenance therapy, because I don't have a lot of data to support this. So that's part of the ongoing research, and that is actually a major barrier because there's not a lot of good metastatic Ewing models, right? So it's the metastatic disease that's killing our children. It's not the primary disease. You find it early, you're resected, chemotherapy. If it's early stage, the chance of survival is very high. It's the metastatic disease that's killing our kids. So what my honest God thought is, once the primary tumor is breaking off into metastatic phases, it's utilizing its glycogen to become metastatic. That's the hypothesis, and I think that's where the therapeutic window would be. I don't want it to be replacing surgery or radiation or that first line of defense. I think it should be a maintenance therapy. I envision it, if there's more data to support this as well, is like a maintenance therapy that prevents future metastatic events in Ewings. So that's one part of the question. The other part of the question is hitting glycogen can augment and boost chemotherapy responses to the tumor. So, chemotherapy actually relies on a lot of oxygen tension and permeability of the actual tumors. So your sarcoma in general is just a solid sheet of tumors. There's not a lot of vasculature. It's highly hypoxic. Is remodeling the tumor by getting rid of glycogen can remodel the tumor itself to make it more susceptible to chemotherapy or radiation therapy? That's another question that we're also very interested in knowing. Hopefully, once we get the lab settled, we can get onto that testing. I think that by far would be the easiest way to bring glycogen synthase therapeutics into the clinics, is to be able to demonstrate that we can use this in combination with already established therapy and give its added bonus of killing the cancer cells and increase survival. Because it's hard, I know a lot of these therapies is not super, effective, but they do work. A lot of these chemo and surgery radiation, they do work on a lot of people. So we don't want to take those proven therapy away from Ewing sarcoma patients. The best case scenario is to add something to improve on the already known therapies to be able to boost their cytotoxicity or efficacy to really, brings to better combat the cancer using the current conventional therapies. So yeah, so two routes that we're really hopeful with this research is, one, as a maintenance therapy to prevent future metastases, and two is to maybe help augment current therapies to help that. And the second one, to help current therapies, that's an easier route to bring it into the clinics to do a combination therapy. I think clinicians would be more willing to do that kind of a test as well, because you're not telling them to take away the already known therapies that they know that works. You're not telling them to stop doing that kind of a thing.
Okay. Thank you.
Great. And then one more question. Are there potent supplements or naturopathic options that reduce glycogen stores that you're aware of?
Ooh, I might have read something on PubMed. A lot of the natural, um, some natural plant substrates does hit glycogen synthase. I can't tell you the exact names or where they come from right now, but I definitely remember reading those on PubMed. There's this relationship between plant and humans because we eat them from evolution, and the plant produces some kind of a chemical in them that affects our glycogen store because they're our food, and modulating our store affects our ability to eat them and how often, how frequent we eat them too. And because what you eat from plants is also glucose, right? You convert all of that starch back to glucose, back to glycogen. So I definitely read literatures out there from natural compounds, but again, all of the natural compounds are not very, one, low potency, but two, they hit other targets in your body too. It's not a specific glycogen synthase inhibitor. I can't make a recommendation to take them. I really can't. But I know they're out there. If you do a PubMed or Google Scholar research, I think you can find them. But I don't know. I don't know if they're not going to work, but I also don't know if they are going to work. I can't make recommendations.
That's fair.
Never personally tested them. I guess if that makes any sense. Yeah.
That's more than fair.
Yeah.
Right. Got a couple more minutes here, Dr. Sun. I guess I just want to ask one final question. Seems like you have a really clear path towards targeting this tumor type. And what is the rate limiting step for you guys moving forward as a lab and really taking this next step and moving it to the clinic?
Rate limiting step. Let's see. Number one, people. I need somebody to move down here to Florida. And same as every other lab is it's everything in the lab is expensive. It runs a $1 million operation with a lab of 7 or 8 people. The annual cost is close to $1 million just from paying everybody's salary, and every a single nude mice, it's like $500-600 you buy them, and then you do a powered analysis where people accept the numbers. You gotta do like 20 per group, and then you inject the tumors. Resources are limited just because we can't do everything we want to do in the lab. Sometimes we have to pick and choose just because there are plenty of research labs that ended up losing all of their funding from going down a rabbit hole and then just collapsed, and the PI ended up teaching, right? Become a full-time teaching professor. That happens all the time in academia. Like, in fact, half of my department is like that. Half of my department, they're supposed to do research, but they all kind of lost their funding and just can no longer do research anymore. It's a sad kind of a thing in academic research. Is the resources and funding. The successful ones, I'm somewhere in between, but the real successful people, they know what to do, what to write, to go for like things that will get them funded. And then you never hear me hear this, but like it's hard to get funding for Ewing sarcoma research, but we've managed to keep going through resources from our other fundings, right? It's easier to get lung cancer research. It's easier to get Alzheimer's disease research, but then there's also institutional support. So you kind of mix it apart and just trying to keep pushing on this, right? This is the reason that V-Scholar and Symbiotic Scholar, those foundations exist as well, is because there's not enough money out there for pediatric cancer research. There's just really not. So those foundations exist as well. So yeah, I say human, just manpower and resources, I think, to really trying to push. But we do have a really good relationship with Maze Therapeutic. In fact, we help them establish their current drug that's going into the phase 1 clinical trials right now, just because they need a lot of validation from their glycogen models. So we've mentioned Ewing sarcoma to them multiple times. So there, you guys are definitely on their in their mind when they're continue to push this through orphan drug status.
Well, thanks for keeping us on the radar and for keeping us in the mix, especially with regards to all of those larger cancers. I think we're out of time, so I'm going to close this out here. I want to thank you, Dr. Sun, for presenting and for giving us your
No
time.
problem. Yeah, thanks for inviting me.
And yeah, from the greater Ewing sarcoma community, just thank you for keeping this a priority in your lab, as you mentioned.
No worries. And if anyone needs, have more questions, just feel free to email me.
Okay. And then just one last thing. Thank you to our viewers for all joining us today as well. We'll see you back here in October. Please be sure to subscribe to our Ewings U email. You can do that by going to littlewarrior.org and navigating to Ewings U tab on the website, and also follow us on Instagram and or Facebook, as we always post about our next speaker that way as well. Lastly and closing, we have a sign off here at Little Warrior that we feel like embodies our fight in honor and in memory of many little and big warriors who have faced this cancer. So Dr. Sun, we're gonna ask you to join us. It's just simply a 1, 2, 3 swords up. So if you will, I know the rest of you are all muted, but the three of us are sign off that way. So 1, 2, 3 swords up, everyone. All right, again, take care.
See ya.